Well, good morning, everybody. Thanks for coming back. Hopefully you enjoyed yesterday's events. And so this is the last day. One thing that I was asked to let everybody know is that, please wear your badges because the security doesn't let some people in. So, and if you have trouble, go to the registration desk and then they'll print you a new badge. One thing that I wanted to mention is that, so today we'll have a keynote talk by Saskia, who is here at AIND Institute, and she's going to talk about the AIND Institute for Neurodynamics data platforms for neurophysiology and neuroanatomy, like how to integrate all this sort of different type of data sets, and within the context of collaborative open science, so which is the theme of the ODAN in general. And then after that, we'll have a short break. We'll come back here for the session for preparing data for multi-scale analysis and AI models. And after that, we'll have lunch, come back here for the afternoon session and last session, which is the computational neuroscience and multi-scale modeling. Today, we don't have any parallel breakout sessions. So then we'll come back here for the, I will do a brief, basically synthesis, like a 15 minutes or something like that. And then we open up with a panel discussion, and the panelists will join us to continue. So let's please welcome Saskia, who has been doing fantastic work here in open science. She has been involved in it for many years. So let's hear what she has to tell us.
Great, thank you. Thanks, everyone. The problem with speaking at the beginning of the morning is I don't get to use the, I'm the only one between you and the rest of the day. So I guess we'll just get started. But at this stage in the conference, I don't quite need to motivate too much why open science is important and why it's valuable for us to be collaborating and working together to solve the challenges of the brain. But just to kind of remind us that our ability to collect data has grown tremendously in the last decade, particularly from investments from the Brain Initiative and other funding initiatives to really advance our technologies for recording simultaneous activity in the brain using a variety of different techniques. And one of the things that we've been discovering is that as we explore the brain with these new techniques, a lot of our initial understandings of how the brain worked based off of single cell recordings and earlier methods, we're starting to call into question the validity of those, how they scale across the brain. At the same time, we're finding that our ability to integrate data across different modalities, and this is a recent review of some of the work here at the AIND Institute, really trying to bridge different types of data, different modalities of data to be able to pull these pieces of information together in order to understand the cortical circuit, how it's organized and how it functions. And we're really in a moment where we need to be leveraging our ability to work with large amounts of data as well as different modalities of data in order to integrate them to advance our understanding of the brain. So here at the AIND Institute for Neuroscience, for Neurodynamics, you've heard of some of the stuff what we're doing, but we're really looking to understand how the brain circuitry and activity drives behavior, learning, decision making. And we're trying to answer this question with single cell resolution, but with across a brain-wide scale. And so I'll just touch on a couple of the things that we've been developing in the last few years that we're trying to use to kind of push forward on these questions. Josh Siegel runs our EFIS team, and their team has been really advancing the NeuroPixels platform in a couple of key ways. The first is they've been working on the development of the NeuroPixels Opto probe. This is a NeuroPixels probe that has light emission sites embedded along the probe that allows them to emit light along the length of the probe, 2 different wavelengths of light, so that you can do optotagging kind of below the surface of the brain, right? Instead of just doing the light on the surface, you can actually now get the light down into the tissue. And so you can see an example over here. I don't know where my cursor is. There we go. Over here, an example looking at neurons in the striatum, the medium spiny neurons, that one expressing D1 receptors, the other expressing D2 receptors, that can be distinguished using these optotagging techniques. In addition, they've been building a modular insertion system in order to position NeuroPixel probes really anywhere in the mouse's brain. And they're doing this so that they can really do simultaneous recordings on a brain-wide scale to look at different connected regions of the brain and to be able to look at that activity simultaneously, record that activity simultaneously in the context of decisions. And so one aspect of that is that it's one thing to be able to position your probes anywhere you want, but you also need to be able to target the brain structures that you care about. And many of these brain structures are very small, and there's inherently a lot of variability from animal to animal. And so they've been developing a method to use MRI prior to insertion, where they collect an MRI image of the mouse's brain. They can then register that to the Common Coordinate Framework, and so they can get really specific targets for that particular mouse, for the particular brain structures that they're interested in. And so you can see this example on the right of a recent recording with, I think, 7 different probes targeting different brain structures, all of which are connected to the medial prefrontal cortex, so that they can really start to think about how this circuit is working simultaneously rather than across different sessions, across different animals. In addition to these developments on the NeuroPixels platform, you've already heard from some of our scientists. Carl Schoonover told you about his work trying to do chronic recording with NeuroPixels across many days and weeks, and to be able to isolate the same cells across that entire time to be able to look at the change in the connectivity between pairs of cells as an animal is exploring its environment and learning. And so you can already appreciate the challenges that poses to advancing these techniques. Lily told you about the myomatrix arrays that now are recording from the muscles for different muscles in the mouse as the mouse explores the motor observatory. 2 more quick examples that I'll highlight. One is a calcium imaging study. This kind of builds on some of the previous work that we've done at the Allen Institute looking at visual detection behaviors in the visual cortex, where an animal is performing this behavior and we can record from several planes in the visual cortex simultaneously. One of the things that we're doing here is we're now trying to image across learning, right? So doing longitudinal studies over many weeks, but then at the end of this experiment, combining this with post-hoc spatial transcriptomics, where we can use MFISH to look at the RNA expression in the cells that we've just done functional imaging on, allowing us to get cell type resolution on the populations of cells that we've just been imaging across the learning of this task. And then finally, a last example, which Karel mentioned in his introduction 2 days ago, recent work from Jeremiah Cohen's group looking at the locus coeruleus. And this is a really beautiful study that combines many, many different pieces of data. But just to highlight kind of 2 pieces of it, one is these single cell whole brain neuron reconstructions. Where we can image and track individual cells. These are 4 example cells, all with cell bodies in the locus coeruleus, that show the compartmentalized projections of these neurons to different parts of the brain. And one of the things that they found is that where these cells project does correlate somewhat with the position of the soma in the locus coeruleus. So the dorsal cells are the ones that are projecting up here to the front, and the ones that are more ventral in the locus coeruleus are the ones that project down to the brainstem. And in parallel experiments using neuropixel recordings of neurons in the locus coeruleus, these norepinephrine neurons, they found different learning signals in these neurons, and I'm not going to unpack this, but during this dynamic foraging task, there's some cells that encode the reward prediction error and other cells that are encoding the lack of reward. And these also line up along that dorsal ventral axis, indicating that the reward prediction error is being projected to the front part of the brain. The lack of reward is being, these learning signals are being sent to the back part of the brain. And we've got kind of 2 different signals in this common system. So my point in showing you these examples is to really give you a sense of the range of different types of science that we're doing that integrate neurophysiology, be it neuropixels or EMG recordings or calcium imaging recordings, along with a variety of different behaviors, and trying to also link that to a number of other modalities outside of the physiology space in order to really pull apart these circuits and understand how they function. And so in order to support this type of science, we need to have a data platform that, first of all, is multimodal, It can support a variety of different techniques, a variety of different modes of data, not just that it supports each one of them independently, but also allows us to integrate across them. We need a platform that's flexible, that we can use equally with animals and data that are doing different types of behaviors, that we can extend it when we start to bring in new techniques and new tools, and that is able to allow people to use these data in a variety of different ways. And finally, we need a platform that's collaborative. You can't do these types of experiments where we're doing all of these different pieces. We've got the single cell reconstructions, and we've got the behavior, and we've got the recordings. You can't do that without collaborating with other people. And these people can be people within our own institute, each one specializing in a different aspect of the experiment. So this might be one of our scientists doing the physiology recordings. This might be somebody doing anatomy, and a third person might be building models that integrates these data. And in order for these three people to work together, we need a platform that helps them to share their data and work in a common space. And even when we're in the same building, even when we sit in desks right next to each other, this is a hard challenge, right? Is being able to actually access the same data and work with it and not have, I have version 1, you're working on version 2, resorted, and then we're all out of sync and everything falls apart. And so we've been building our platform using a cloud-based platform where we bring all of our scientists into the same space and bring our scientists to the data rather than trying to distribute our data to everybody independently. This is really important both for ourselves when we all sit on the same floor, but it really becomes more beneficial and more impactful when we think about when we're trying to collaborate with people that aren't in our building, but collaborators who might be across town or on the other side of the country or in another part of the world. People who might have different be bringing their own data that's relevant to our questions or have a different modeling approach that they want to use to try and understand these data. And by bringing them into the cloud, it really makes it much easier for us to quickly share the exact same data, the exact same analyses that we're working on, and keep that group of scientists kind of working in sync. Ultimately, though, we're trying to build a platform that supports open science. That's not just for our friends and families, the people that we are already talking to, but we want to make it possible for anybody to be able to access and work with these data in the same space that we are. And so the platform that we've built leverages the cloud to bring scientists around the world into that same space, being able to contribute into using the data, analyzing the data, and helping us to even potentially build tools and build things around this. All right, so our platform is really centered on this idea of community collaboration. And so this means that there's a couple of things that we need this platform a couple of properties we need it to have. The first is that it needs to be accessible. All parts of the workflow need to be openly available. And so this means that we put our raw data into the cloud pretty much as soon as we collect it. The only thing we do before we upload it is compress it so we can save on storage space. And then everything else happens in the cloud. Our data processing, our analysis, all of that lives in the cloud. Any derived results, the NWB file with the spike sorted units, that lives next to the raw data. All of that entire provenance chain is open and accessible. We need our platform to be standardized. Data is immutable. We can't kind of overwrite files underneath people while they're working with it. And so when data gets uploaded, that file doesn't change. Now, I might reprocess my spikes. Kilosort 4 came out. I processed this earlier with Kilosort 2. Now I want to try Kilosort 4. I'm not going to overwrite those previous spike sorting results. I'm just going to create a new derived result with the new processing, but keeping track of the provenance so that I know the difference between these two. In addition to it being immutable, we wanted to conform with standards because having standards, leaning into standard file formats and standard tools allows us to build tool chains that we can use across different experiments and across different contexts. We need it to be transparent so that people who come into the platform are able to find and understand what the data is in order to facilitate them reusing it effectively. And we need it to be reproducible. When we build pipelines and analysis workflows, we need those to be something that when we share them and when we reuse them, we know they're doing the same thing. And so we lean into a method of containerized compute that allows us to establish these reproducible analysis workflows. So to kind of walk through how we've implemented these 4 principles, as I mentioned already, all of our work is being done on the cloud. We put our data in an open bucket on AWS. We upload the data after pretty much as soon as we collect it, and then we keep all of the processed and derived results in the same bucket as the raw data. Our code is all in public GitHub repositories. We also have a lot of protocols on protocols.io. All of this is open and accessible. We lean into standards for our data formats, NWB for our physiology, OME for the light sheet imaging data that we're acquiring, as well as a data standard that we use for our metadata that I will discuss more. For transparency, we've been building, one of the key things here is we've been building a metadata catalog, leveraging our data schema that allows us to keep track of all of the data, where it came, how it was collected, how it was processed. That is then something that anybody can query in order to find things or to understand what a particular asset that they're working with has, as well as building data portals to be able to, again, explore the data and dig into what it contains. And then finally, for the reproducibility, I mentioned we lean into kind of this containerization, leveraging a cloud-based analysis platform called CodeOcean that is really designed around this concept or this philosophy of reproducibility. So briefly, to discuss this a little bit more, the key thing for reproducibility is we need to understand exactly how data was generated. This both includes how it was collected, but also any processing that happens. When you have a derived result, you want to know everything that happened after that processing. And so keeping track of that data provenance allows us to track anything that's happened from the raw data to the results. And having shared tools that work the same way. In one of my breakout groups, I think on the first day, we talked about kind of one of the challenges is that we're all kind of creating the wheel over and over again in terms of building up spike sorting tools or video processing tools. And part of that is because a lot of these tools, like it does most of it, but there's a lot of pieces that go around it. And so we really need to build these robust pipelines that we can use really as a pillar rather than as a piece of the puzzle. Kind of to illustrate this, we can talk about how for our pipelines, if you look in the methods of many papers in how the data was processed, you'll see something along the lines of we use Sweet2P to process our data. The authors aren't doing anything wrong here. They did use Sweet2P. This is a complete accurate statement, but it's very much like this instruction on how to draw an owl. You start with 2 circles, and then you get an owl. And there's a lot of information that's actually missing about how they actually process the data. And so because of this, we need pipelines that go beyond just we used this common tool, but show the full array of what that entails. And so this includes all of the precise steps, the pre-processing steps, adding in the parameters that get used, and how these things are fine-tuned for particular things. So CodeOcean, I mentioned already, is a platform that we've been leaning into for this containerized workflows. They're built around these concepts of capsules. A capsule combines code, data, and environment settings. When you have all of these 3 things contained together, every time you run that, you're going to get the same result. And one of the things, none of this is magic, right? I've shown here this. The environment is defined using Docker. The code is synced with GitHub. These are all tools that we've all heard of. But many of the scientists in our institute, yes, they've heard of Docker, but how exactly do I use it? How exactly do I set it up? And so the thing that CodeOcean makes really easy here is it has a no-code GUI for creating these capsules, and then we can share these capsules. And so the scientists don't have to become, computer scientists. They don't have to become real sophisticated software engineers to be able to really use these quickly and effectively to share their work with collaborators. There we go. In addition, when we run data through capsules and save results, CodeOcean creates a nice provenance map. So for this derived result here, it shows me which capsules were run and what the raw inputs were to that capsule. And I can then kind of go in and explore within this capsule what exactly the code was, what the conditions were, what parameters were set. And so it has this built-in provenance tracking so that we can keep track of the work as it moves through these workflows. And then finally, we can link these capsules together to create pipelines, where each capsule has different environment settings for its particular need, but we can string the outputs of one capsule into the next capsule. Some of these might be pretty simple steps, others might require a lot of compute, and so we can scale the compute accordingly across the different capsules, and use this to create full pipelines for processing data, for instance, from our raw ePhys recording into an NWB file that has all of its the spike sorting and time synchronization and stimulus information all integrated into that file. So our teams have been building a number of these pipelines, one for our NeuroPixels ePhys recordings, another for 2-photon calcium imaging, another one for some of the light sheet imaging that we're doing. And one of the things I'll point out is that these pipelines are largely built on community-based open source tools, right? So our spike sorting leverages Spike Interface and Kilosort 4. Our calcium imaging is using Suite 2P and Oasis. Both of these are putting data into NWB. These are open source common tools. We're not trying to build a whole new way of doing this, but it also has custom tools around it for doing those pre-processing steps, getting quality control assessments built into this process, and kind of combining and collecting some of the metadata that we need to consolidate as well. All of these pipelines are open, accessible pipelines. So the CodeOcean, we can string these capsules together. It ultimately creates a Nextflow script. Again, you don't have to be a Nextflow expert to be able to build it, but it does create this Nextflow tool that we can then share with other people. So all of these 3 pipelines that I just showed you are on GitHub. You can go to these QR things to find them if you're interested. And they can be deployed in any, you can deploy them on your local machine. You could use them on your Slurm cluster at your university. There's instructions in the repositories about that type of how to do that local deployment. So they are accessible tools that we've been sharing with other collaborators in the community. All right, so the one key that I mentioned now is this reproducible workflows that we can use to keep track of the provenance and have access to the full workflow of our data. The other piece that I want to come back to is the importance of our metadata and file standards. I'm kind of excited that I don't have to convince you too much about metadata. We've been talking about it already in this meeting. I think often I'm in situations where I have to make that hard sell. But just to reiterate, the standard files, standard metadata make our data more accessible and usable and allow us to build tools that we can share across different experiments and across different teams. There are existing standards, and I said we lean into these for our data organization, but many of these ultimately fall short on the data documentation and the metadata. And so we need a system to have that metadata that allows us to contextualize and formalize the embodied knowledge that exists about the data from the people who collected it, that will allow users, be it their lab mate who's building models or somebody around the world who they've never spoken to really understand what that data is to reuse it. And just to kind of highlight, again, why this is important, if we go back to the FAIR principles, findable, accessible, interoperable, reusable, if you go look at the paper, there's this box about these principles. They've kind of broken them into bullet points. And basically every single bullet point calls out metadata being an important piece to make data, to have data actually be fair, actually be open and reusable effectively. Another kind of example, a recent paper that looked at a replication of a number of AI studies, looked at how well they could reproduce these different papers. I think they found they could reliably reproduce about 50% of the results from an assortment, I forget of how many, 30, of 30 different papers. And one of the things that they call out just even here in the abstract, is that the quality of the data documentation correlated with successful replication. So being able to actually understand what the data was enabled them to be able to reproduce the results of the studies. So the schema that we've been developing in neural dynamics allows us to kind of track the data, all of the features of the data that we've collected. So it'll start with describing the subject. All of our work is in mouse, but it could be a variety of really any species, but we want to know the species of the subject, other features such as the genotype, the sex, the age. For our mice, we want to know some information about its house, particularly its light cycle. We were talking about the importance of time as a variable in experiments, so knowing these features of our subject prior to any experimentation. Then we want to keep track of procedures that have been done to the animal before we collect data. So in this example, this mouse has had some stereotactic surgeries. This includes some virus injections. These viruses were injected in a particular coordinate targeting a particular structure of the brain. The virus that was injected is a particular virus. It has a plasmid, and we want to know what that plasmid is. We also want to know which stock of plasmid of virus this is, so that if we need to at some point look for batch effects across different batches of virus, that is information that we have access to. This mouse also had a fiber optic implant. This fiber has some optical characteristics, and it was also implanted in a particular location of the brain with particular coordinates. And then this mouse also got some water restriction because we're going to use it for behavior experiments. And so we want to know which protocol of water restriction was used, when did we start this, and keeping track of that information. When we go to collect data, we want to know what instruments we're using. So for our fiber photometry, we're using some lasers. There's some behavioral equipment that the mouse is using. In this case, maybe it's just a running wheel, but there might be other devices as well, a monitor or olfactometer. In this case, we've got some cameras that are tracking the animal's behavior as well. And then when it comes down to actually the data acquisition itself, for each asset, we want to know what were the parameters and the settings and configurations of those devices that are being used. What was the power of the laser on this day? What were the parameters of the behavioral task that the animal performed? And so we can keep track of how the data was acquired. And then once we have this raw data, this, of course, gets uploaded, but we might then start doing processing. We're going to do signal extraction, temporal alignment. And so we want to keep track of each step of that processing. What was the code that was used? What were the parameters that were set? And what outputs did it create? On top of these things, there's two other important pieces of information. One is the data description. This is, who collected it? Who funded it? Where did it come from? I think oftentimes when people talk about metadata, these are the things that they are talking about. These are important pieces of information for finding and knowing what data belongs to who and where it came from. So it's important that we have it here, but it doesn't tell you actually what that data is, right? It gives you kind of that administrative context. But the last piece that we have in our schema is the quality control. Where we can put in evaluations and annotations of the data that's been acquired, right? So as we're uploading our data as we acquire it, there can be experiments that aren't great, right? That as we start to do our image processing, we find that the signal-to-noise ratio of these movies is really, really low, and our algorithms aren't going to detect cells. And so when we might end up not analyzing and not using it. And so we can indicate that in our metadata. And so what we found is that the signal to noise of our calcium movies was really low, but what was great were the behavior movies. And so we're going to say, the calcium movies are bad, but the behavior movies are great. And so if you're trying to do an analysis on behavior videos, you don't care about the calcium signal, this is still valuable data that you'll be able to use. And so having these evaluations on different pieces of the data allow downstream users to be able to make informed decisions about which pieces of data are reliable and valuable for their analyses. So this example is an example of a fiber photometry experiment, and as you know, we do a wide range of different experiments, and so it's important that our schema be able to capture the details of different types of experiments, right? So in addition to fiber photometry, it could be a neuropixels recording, could be calcium imaging, could be, in this example on the bottom, experiments with our light sheet imaging, where we, after we inject a variety of viruses, we're perfusing the brain, we're clearing it and expanding it. We're using our expansion assisted light sheet imaging. We've already applied some antibodies. We want to know what those are. And then we can use the light sheet imaging to collect these in vitro imaging data sets. And so we need a schema that handles both of these different types of modalities of data and allows us to have points that we can share in common across them so that we can best integrate them. And one of the tools for this, the schema itself is designed for this, but in addition, we lean in when possible to persistent identifiers, public databases, and ontologies in order to annotate key pieces of information with these types of annotations, which become really crucial for communicating both with other people what it is we're doing, what the tools are that we're using, where in the brain are we targeting, and so communicating that to other users, but also becomes that really important linchpin for bringing data across different modalities and being able to integrate the data in these ways. All right, so this is kind of what the schema is. Around the schema, we've been building a number of different tools. If you talk to kind of pretty much any scientist here, they'll tell you metadata is really hard. Getting these complete, accurate documents of the data is a hard challenge. You need to do it from the get-go. It's not something you can do retroactively. And so we've been working in the scientific computing team building tools to facilitate this, particularly for our team. So one is a metadata service which works with some of our internal systems where our, the lab animal services who maintain our animal colonies, they have their own database for each animal. And so we can just pull our metadata from there rather than asking a scientist to go look it up and transfer it into the system. In addition to that, we have a GUI for metadata entry where users can actually go in and can fill in fields for their particular class of the schema, and work closely with our scientists to develop mappers for their particular systems that work with particular acquisition software or particular kind of configurations that they have within their team to try and streamline this process of capturing the metadata from the scientists as they're acquiring the data. So as we're able to ingest this data, it gets uploaded with the data. It lives alongside the assets in our S3 bucket, but it also goes into a document database. So for each data asset, we have a JSON file. I think I forgot that step of it. The metadata is in JSON. And these JSON files live in a document database that then can, where we can go and find each record, we can query it, and we can see kind of all of the things that are happening. On top of this, I want to tell you about two tools that we've been developing. The first one is a QC portal for quality control. This is really one of the only things that can get added into the metadata after it's created, is these quality control evaluations and annotations. Because this requires somebody to actually kind of look at things and make decisions. Some QC can be automated, but a lot of it you need somebody to at some point go in and make an assessment. And so we've built a portal that allows us to access information about each record to make these assessments. So an example, so this is just showing you the portal. Each row here is a single asset. If we go and look at one of these assets, you can see kind of visualizations that allow users to make a judgment about different features of the data. And so just to kind of walk through the different parts of this, in the top, it has a kind of an overall state of the QC for this asset. As I mentioned before, there's going to be different evaluations for, say, the calcium movie and the behavior videos. And so we want to kind of keep each evaluation separate so you can see, you can assess the value of the data for different needs. So the top view there shows what are all of the different possible assessments and what is the status for this particular asset for each of those things. Within them, there's, again, a list of different evaluations. And for each evaluation, there are a variety of metrics. And these metrics usually have some sort of reference image that the user can use to make that assessment that hopefully has done some processing and kind of contextualization that you can then look at it and easily make a decision, is this good, is this bad? And these types of visualizations can really, can be all sorts of things. They can be static images, they can be videos, they can be neuroglancer links that users can interact with. And then finally, approved users are able then to actually go in and make assessments and submit these results through their account. So just a quick demo of this. You can see here's a list of all of the assets in a particular project that we have. You can see lots of different assets. Each row, again, is one of these assets, and the overall QC status is indicated in that column. And here, looking at, this is an example with neuroglancer links, where you can actually zoom in, move around, you can turn different channels on and off in order to assess this evaluation. This particular one is overall tissue quality. This next one is looking at the output of a cell segmentation algorithm, and so you can actually click on and off the cells that were identified and compare it to the image underneath it. And the last assessment here is, or evaluation here, is actually an annotation of, in this case, there were injections, and we want to say exactly where we think the center of the injection was. And so that's a manual annotation that can get entered in this system. So this information, this portal kind of will write back into the document database so that that information now lives in the metadata record of each asset. So coming back to our document database, we've got lots of records. I think, right now it's something like 40,000 different records. So if you want to find information for a particular data asset, for a particular project, you need to be able to query into this document database. And so if you know SQL, if you know MongoDB, you can easily write these types of queries where you pick a couple of features. So I want to find all of the assets that are part of a particular project that have had a type of injection and that had a quality that passed. I want to access their overall tissue quality evaluation so that I can use that to select particular things. And there's certain features of these assets that I care about, a lot of other features I don't care about. I don't actually care about what instrument was used to image this, but I do care about what virus was injected, where it was injected, and the age of the animal at that injection, right? These are the things that I want to build my analysis off of. And so I can build this type of query. I can make a pandas data frame. I can put it in my notebook in a capsule and run my analysis. But usually when I talk to people about this, I say, if you just use MongoDB, it'll be great. And I see eyes just widen, because it's oh, that's another thing I have to learn. And so one of the tools that we've been developing is a large language model to interact with this database. So we call it Gamer for generative analysis of metadata retrieval. And this is a chatbot that uses kind of a number of different steps using large language models. One branch of this is that it will create these MongoDB queries. So you can ask it to retrieve something, it'll create that MongoDB query, it'll execute it. Or it might you can ask it just to give me the MongoDB query so that I can just copy and paste that code somewhere else. It also has an arm where it's done a vector embedding of all of the assets in our database, and so it can use that type of approach to find particular records very quickly, relatively quickly. And then finally, we have a similar embedding of the actual schema itself, so that you can ask questions about the organization of the schema and the structure of the schema through the chatbot. So a couple of examples here on the left. Find the assets in this thalamus in the middle project where GFP was injected into the target structure VM, list the subject IDs. And so it shows you a little bit of the logic that it used on using MongoDB on the database. It comes up with a list. I think there were 24 things here. I think one of the things that I like that I want to underscore is that the virus is never GFP. It's always AAV-PH with some GFP off in the end. And there's 3 or 4 different variants of GFP viruses that get injected. And so, the language model clearly can do that abstraction and get all of those different variants together. The second example, again, what are all the brain structures injected in the thalamus in the middle? It pulls up the names. It actually can figure out what the structures were. I think it only has access to the acronyms. But here, it actually has printed out the Python code to reproduce this. And so if, it's one thing to get the answer to this, but if I really want to integrate this into my analysis workflow, I don't want to have to copy and paste this list of structures into the next thing, but I can take this code, that's the first cell of my notebook, and now I have this list of structures, and I can use that from there to go further. The last example on the right, tell me about mouse 747107, and you can see it gives a nice summary of this mouse. Mouse 747107 is a female mus musculus with the genotype blah, 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 blah. It underwent planar optical physiology using the mesoscope setup. It was imaged with 920 wavelength at various depths in the visual cortex, ranging from 68 to 260 micrometers. It's giving a nice narrative of the experiment that was done here. And if I'm writing a paper just about this one mouse, I've got my methods section ready to go. All right, so just to kind of come back to this picture, right? So this is the platform that we've been building around community collaboration. The reality is that each of these pieces is modular, right? I told you about our pipelines that you can actually just take and run on your own local machine. You don't have to use them in our platform, although it's really easy and nice to use them in our platform. You already know about data, our standard data formats from the community. These are all tools that everybody can use kind of in a mix and match kind of way that would be useful for you. But at the end of the day, I think that the real power comes from bringing all of these pieces together. I think one of the driving motivations for the design of our platform is that we're trying to move away from a form of open science that's built around dissemination, where we collect a lot of data, we create a bunch of resources, we create a bunch of tools, and then we distribute them and disseminate them into the world, and that anybody can then just take them and use them on their own. I think that's great, I think that's wonderful, but I don't think that that's going to be the thing that moves our field forward as effectively as if we can actually bring everyone together to work in a common space and sharing tools in a way that we can actually build on top of one another's work. And as we continue to develop this platform, we really want to be driven to develop an architecture of attention that helps the people with different types of expertises, different areas of interest, to be able to plug into this, to contribute to those different pieces so that as a whole, we're able to leverage all of the different work that's happening throughout the community, with both on an experimental level, but also on the modeling and analysis level, in order to really drive forward our understanding of how the brain works. So with that, I will wrap up. I want to thank everyone on our scientific computing team who's done a lot of the work on building this infrastructure, as well as many of the scientists who built some of the examples that who've collected a lot of the examples that I've shown you at the beginning. And I'm happy to take a few questions.
All right, Vincent. You can yell. Okay.
Thank you for the nice talk. I have a few comments. I have some comments about what could be the remaining roadblocks for adoptions for all these tools. One is so I love this idea for the gamer software to retrieve data that is already metadata. I'm thinking about the input side. When you tell people, and from experience, this is basically what type of metadata you will need, to put your data and share it. Usually they are overwhelmed, because they are like, oh, that's too much. I don't know how I'm going to do that. And, or I'm not going to do it. Sometimes you have that reaction too. Would it be possible also to have, this kind of chatbot or whatever, some kind of interface that says, hey, ask me anything, and then people say, what information will I need to put in, and how do I put in this JSON file, and then it uses all this code-based knowledge and so on to generate either the script or use whatever the user has.
Yeah, no, I think that's a great question. I mean, you really zeroed in on exactly the challenge is that metadata capture at the get-go. And we can build tools that work well in this building because we already have some infrastructure in place, but those aren't necessarily useful are not at all useful for people outside of the walls. And so this is one of the key things that our team is really working on at this point, is thinking about what are some ways that we can reduce that barrier for metadata capture and the use of large language models as a way to kind of guide that process, right? Streamline that process. What I don't want is a chatbot kind of making up metadata, but if it can kind of guide somebody through that process of what are the key pieces of information, what information do they already have somewhere else that they can just pull and grab and go, and really kind of streamline that process. I think is definitely a direction. And I think just building more kind of custom tools that, not custom, but tools that are kind of specific to, again, shared resources, right? If there's acquisition systems that are being used broadly in the community, if we can build tools that work nicely with those acquisition systems, that can then be leveraged kind of throughout the community. But that's exactly one of the key things that we're thinking about and working on, yeah.
The other comment on adoption is for the pipelines. So these are great pipelines. I actually used the EFIS pipeline. I set it up. It was working pretty well on the local cluster at my institution. But it is not exactly the same setup that you use, because you use Codation, you have AWS data buckets, and so on. And I know a number of labs that would not use these tools, or not all of those tools, because they have to pay for them. What do you think about well, first of all, I think for all when you present this thing, it should be. I mean, it would be nice to have also an estimate of how much it would cost, for a given lab if you wanted to adopt the whole thing. But also, if you don't want to use AWS plus Codation and so on, but you can still run it as I've done, to explain how that can be done, and maybe having a GUI also, that the same thing as Codation, but something that helps you interact with this nice capsule, chains.
Yeah, I mean, I don't think that we can build a GUI that does the same thing as Codation.
Not to that extent, but
then, yeah. But no, these are all fair points, right? There is adoption of a tool such as Codation, does require kind of some choices, right? And it does require some, using the cloud does have a cost associated with it. I will point out Codation does have academic pricing models, so that for academic labs with, I think, less than 25 people, the licenses are free, and so you're really just paying for the compute, paying the cloud cost as opposed to the Codation access. And so that is one kind of step in reducing it, but cloud compute does cost money. And that's a reality, right? But I think it's a reality we have to kind of really think about it, right? Because moving data also costs money. Storing data on, if you're going to take all of the data that I'm collecting and get it over to your institute and build a bunch of hard drives, all of that is still a cost. And so this is just moving that cost in a different place. And I'm not trying to diminish this, but I think it's an important piece for us to recognize that this is true of any system that we build, is that there are costs associated with it. But I think the key thing, though, is that that moving and distributing data, the fragmentation of that system, I think, creates a bigger barrier that we're kind of reducing by eliminating, by bringing things closer together, right? If you are going to rerun spike sorting on your own because you got the raw data from me, then you're redoing that processing, you're restoring a new asset, and that also is kind of an unnecessary cost as well. And so trying to reduce the redundancy, I think, is one thing. But you're absolutely right. There is that component, and I think it's a fair thing to talk about in terms of this model.
Scott.
So many of the pipelines you showed went from raw data to NWB file with signals of interest, and I'm curious if you could just say a bit about what the next steps of analysis look like in this framework.
Yeah.
A lot of exploratory analyses maybe are kind of quick and dirty things that you don't necessarily need to consolidate forever, but some of it you do. So just speak a bit about what that looks like.
Yeah, absolutely. I mean, I think you already kind of started, right? Some analyses are, I'm just getting to understand this data, I'm testing a few things out, I want to try different ways of working with it, and that maybe doesn't need to get elevated in a high level. I think particularly with projects as they mature, though, you do want to build an analysis infrastructure, an analysis architecture, so that for this particular project, maybe for a full, a modality, or for like a particular discovery project focused on a particular question, all of the data was analyzed, after it was processed, was analyzed in a consistent way. And so you can kind of leverage the same concept of these data analysis pipelines, like the spike sorting pipelines, as an analysis architecture. And I think using a similar idea, right, where you have some common steps that are basically finding the right assets, attaching them to capsules, kind of indicating which part of the data goes into this workflow, which part goes into that workflow, and then taking results and consolidating them. And in fact, many of our teams are starting to build these types of analysis architectures for some of these more mature projects where every day we're collecting new data and we want to get it analyzed, we want to get the results integrated into kind of a consolidated representation. And so again, leveraging that containerized workflow allows us to build that type of infrastructure.
Carl, did you have a question?
Yeah, I have a comment about the cloud stuff, right? So compute is never free, right? It's done on a universal cluster, and it might look free to you. Free for us. Well, but all of this, I think that model might also change. Yeah. I think it's holding actually academic community back now. I mean, that's maybe a different discussion, but all of that stuff, most is covered by indirect costs. You know that is definitely going to change, and so you will pay for it from your grant, and that's probably the better way of doing it. That you hopefully, right, indirect cost stuff that's saved there will go directly to your lab, right? That's the optimistic outcome. But you see the bill, make a choice, make an outcome, much, much better. That is to have all the jobs as much as possible. And if you really want to collaborate with lots of labs and pulling data across lots of labs, it is very, very hard to do, if not impossible.
Okay. Let's thank Saskia one more time for the